And thank you so much to the conference organizers and to my colleague, Dr. Marcel Perret-Gentile, for the invitation to join you for my first LabRoots event. I wish to start with the disclaimer that the views represented here today are my own and based upon many years of reflection and discussion with valued colleagues that are veterinarians, animal research staff members, IACUC professionals, committee members, and scientists. Dozens of people before me for dozens of years have been discussing variations of what I will share today in the hope that we continue to keep our lines of communication open to better understand our own as well as others' perspectives. So just for some overview today on what we're going to cover, the presentation will have some learning objectives. I'll share the background on the intersection of art as a perspective on evaluating the 3 R's. We'll also review the classic 3 R's for animal use and welfare and bring in some historical perspective. We'll touch on limitations of the rigid interpretation of the 3 R's classic concept and how to think about analyzing animal research ethics with the progressive 3 R's lens. And lastly, if time permits, I'll touch on a couple of case scenarios that have just been presented in the last month, January 2021, of how we maybe can apply these progressive 3 R's. So I have to give a little bit of background on why I decided to bring art into this notion today. So during the pandemic, like many of you, there was a lot of time I gained back with the loss of other activities. And in that time, I found the Barnes Art Museum that had online courses, and I enrolled in a few of their lectures. I never really had time to study art before, and I learned about the collection that's at the museum and about painters like Seurat, Modigliani, and Van Gogh, who I'll highlight today. I was still in the midst of these art classes when I was reached out to by the LabRoots group and asked to participate in this conference. And it was known to me that it was going to be about the 3 R's. That's what Marcel had shared. And I did agree to talk on the condition that I could discuss the 3 R's more philosophically as a critique and a conversation, really, with the audience. As I thought about the framework of the 3 R's juxtaposed against my ongoing art classes, and the frames that I was seeing from artistic masterpieces, these ideas started to merge in my brain. So when analyzing animal research and the cost or harm to benefit considerations, the viewpoint is widely dispersed depending upon whom the observer is. Are you a member of the public? Are you in the media? How about an anthropologist, a statistician, a veterinary technician? Each of these stakeholders in animal welfare will come to the 3 R's with their own perspective, and no one person can really make the determination about that harm-benefit analysis without further discussion and understanding of multiple perspectives. We are part of a scientific community that has the privilege in our country to deliberate on moral issues, and we have the responsibility to keep objective facts and subjective feelings in balance when ethical considerations are involved. We each may see things through what you see on the slide here, these different frames, depending on the scenario that's presented to us. However, I feel that keeping things boxed into just a frame, like the term framework itself, potentially inhibits our ability to think more broadly about the balance of ethics, welfare, benefits, and science. So in looking at these frames, it struck me in my art classes that it looked a lot like pictures of the French Salon. Now, I hadn't heard of the French Salon before, so if you'll give me a moment. It was held in Paris. It was a state-sponsored huge art exhibition held annually with all Academy-trained artists. It was the official venue for displaying the styles of the time. It was held in Paris beginning in the 1660s and arguably was the greatest annual art event in the Western world between about 1750 and 1900. So in my analogy, if you'll indulge me here, we are this audience in the picture today, on the floor. We are the scientists, the animal care workers, the public, the ethicists, the regulatory overseers. We're all standing, looking at the walls plastered with this art. We're looking at the art of science, the art of animal welfare, the art of the species that we interact with. Our perspective on how and where to look in this grand room really depends on where we can get our best views, and it differs on where we're standing on that particular day. So my point here is that the 3 R's are not stagnant. They're not just two-dimensional. They demand continuous review, perspective, discussion, and evolutionary interpretation. So before we go much further into this presentation, I just want to remind everyone about the great number of discoveries that have happened with the benefit of animal research. And this is, I just selected a few from the SBR website on all of these, which have been granted the Nobel Prize in Medicine. They've preceded and occurred after the inception of the 3 Rs, which occurred in the mid-1900s. All of these included animal models for the discovery. And if you want to see more information about them, certainly please do visit the SBR research site. As recently as last year, at the bottom bullet here, CRISPR-Cas technology received the Nobel Prize in Chemistry. It's a technology that will have huge impacts and already has on animal models that we use. And I'll share a case study at the end that involves CRISPR. So just very briefly, since many of you know this, but I think it never hurts to just revisit it, the origin of the classic 3 Rs, as I'll term them. So these were established by Russell and Birch. They were British scientists that were writing a book. And in 1959, they did state that the scientific excellence of what would come from work and the humane use of laboratory animals would be inextricably linked. Their aim at the time was to improve animal welfare, with the 3 Rs simply as a launch pad for the future development of a discipline that they were looking at, thinking about new scientific methods and medical discovery. So what has occurred with the 3 Rs listed here, replacement, reduction, and refinement, is that they have been increasingly applied as the penultimate ethical benchmarking tool against which high-quality science in the academic and industrial sectors gets measured. And there's increasing focus on thinking about alternatives to using animals at all. However, it's not really all that they represent. And it's important to recall that there was never a mention of alternatives in the initial work by Russell and Birch. The concept, in fact, remains more prevalent than ever. So the 3 Rs just has grown as really kind of a branding in global discussions related to humane animal use and aspects of research and teaching and testing. But I want to point out that they're a touchstone in terms of refinement, reduction, replacement. They're not the end-all be-all, and I'll have us think a little bit more progressively as we go through the talk today. So why would they be challenged? We know that they are a contribution to the evaluation process. We can appreciate they are not the sole ethical consideration for animal welfare, but understandably at times they have been. And so I'd like to kind of, in the idea of thinking about them in a pictorial sense, show them as an umbrella. So this would be the 3 pieces. There's 3 leaves here of the umbrella, and there's one hand holding it, the one perspective of the person that might be looking at that. So I want to think about more broadly, if we have the 3 Rs, and here they are again from before, like what would happen if we actually put them in each of these umbrella angles to think about them. As I've mentioned, it's a lot in the wording. So at the times that I feel that 3 Rs have been misappropriated, it's as if one of the Rs has been pulled out and really focused on, and somehow deemed that we maybe are, as a scientific biomedical veterinary community, not accomplishing things and not progressing if we aren't accomplishing a 3 R in each of our experiments in some way. There's an idea that we might be failing, and I object to that strongly. I mean, it's sort of like we looked back and these were coined in 1959, right? So we wouldn't expect an infant, if it was born, to predict its future life course. So we can't really continue to hold the 3 Rs in their nation state. If we accept that that concept has matured and they're really kind of past middle age now, quit the acronym, but it represents so much more to the benefit of ongoing scientific discovery. So for example, it's not necessarily an accomplishment for human benefit if we use fewer animals in the spirit of reduction when we have more disease and illness. And it's not a failure if we have to look at more than just one sex, which again would be more animals, male and female. And it's not a catastrophe if we don't replace animals because we have to have an intact body system and measurable responses for observations, interventions, and outcomes. I doubt there's anyone that would refute that the classic 3 Rs definitions need to be expanded, and certainly we have to realize there's been a massive transition in how the world views science, discovery, and animal use since their creation. So the 3 Rs we'll hear often referenced to as an ethical framework. but I would ask you to think about them just as a tool, one of many, that should be used by the scientific community to assess the cost-benefit, also called the harm-benefit, of using animals in scientific discovery. So back to thinking about our art, if we put a tick of the 3 R's up on the screen here, we can overlay them. And in doing so, I want to share with you a little bit about a triptych here, another Van Gogh piece. So this is a postman. It's very famous. Many of you will probably recognize this. One of these hangs in the Barnes Museum in Philadelphia right now. For most of 1988, Van Gogh rented a room in France, in Arles, near the train station. And he saw this gentleman at the local pub. This is Joseph Roland. He was a mail handler. They became good friends. And over time, Van Gogh painted him multiple times, always with that dark blue uniform of the Postal Service. But what he was able to do, and I'd like to draw as an analogy, is he took what would have been an ordinary kind of salt and pepper beard and turned it into this brilliant ocean of color. So if we think about paintings as a map, the 3 R's are also a map. They're a guidance to us. They're not set in stone. Each time we might look at them, there'd be a different perspective. And we think about them much like art. This is a timestamp of what was going on in France in the 1880s. Just like the 3 R's, we have to look at them and place them in context of what's happening right now, what our contemporaries are doing. We think about them with respect to social problems and social changes and other things that are present and ongoing. I have a great quote by the American artist Jeff Koons, who says, "I'm going to ask you to be courageous, confident, and not think of art as the finished product, but as a process of becoming something vaster." And again, I think there's great analogies here with the 3 R's. So we then transition the 3 R's. I'm going to have you go back to the umbrella analogy with one person holding it and open up the umbrella. So now we have a lot more space in which to work. We're not restricted by that framework that seems to be a very common concept and discussion. And I also want to point out that under this sort of expanded view that gives us much more space to think and work in, we have more than one hand holding the handle. These are all the stakeholders. We need to be having group discussions about the 3 R's to rigorously evaluate the science, the proposal design, the species, the number of animals, outcomes, and impacts. So there are a number of additional important concepts that appeared after the 3 R's. The 3 R's really didn't even take root until the 1990s, honestly. But in that time, from 1959 and onward, I just wanted to give you some more of the synergy that comes from what the 3 R's started. And the 3 R's themselves, to be honest, we don't have time to go into all the history, but they came after a lot of other discussions about thinking about animal welfare. So the 5 freedoms, many of you have heard of them. They outlined 5 aspects of animal welfare that are under human control. And they were developed initially by the UK government in response to livestock husbandry. They themselves didn't get formalized until about 1979, when animal welfare groups in the UK and the Royal Society for the Prevention of Cruelty to Animals started talking about them more so. An additional concept was that of the 3 S's. I'm really grateful. So these initially were coined by Carol Newton, and they weren't widely embraced either until the 1980s. So Carol was a scientist, and she presented this idea at an ILAR conference in 1975. And they really were only presented in a talk. They were later discussed by other scientists, and it wasn't until 2016 that Dr. Smith and Hawkins decided to write them up again. So in the concept that she shared of good science, this is naturally the aim of all scientists. Still is, was then, and will be. So in essence, we're talking about experimental design to reduce variation, permit effective analyses, and obtain the most information with the greatest certainty in the shortest amount of time. Her idea of good sense described an essential tool in lab animal science, in that the right animal is used for the right reason. This is another 3 R's, the right animal and the right reason, that was coined at a 1970s AALAS meeting. So when considering animal models, Carol cautioned that researchers have to critically reflect on the models and the approaches that have been used, and challenge themselves as scientists as to whether the animal use is valid, if it's translatable, if it's justified. I mean, again, these are all terms that she was using well before we use these in our common vernacular today. And lastly, good sensibilities. This refers to the capacity to respond to or be affected by emotions or events. Empathy for our research animals is a prerequisite to the reduction of suffering, and what she called the life worth living for the animals. Now, in today's terminology, sensibilities has largely been replaced by the term sensitivity or care, or the culture of care. And the paper that Dr. Smith put out reminds us that this concept of culture of care aims to improve animal welfare, but also improve the condition of our animal care workers. Finally, good sense, good sensibilities will promote good science, and that's ultimately what they hope to talk about. So next, I'd like to move on to, historically then, the next thing that came along was in 1985. And these were the US government principles. The large title is the Principles for the Utilization and Care of Vertebrate Animals Used in Teaching, Research, and Training. And these were published in May of 1985. And they were part of an ongoing change that was happening even with the Animal Welfare Act for amendments in around that time of 1985. So I'll just kind of step through these with you. The idea behind them was that we needed to develop the knowledge necessary to continue to improve the health and well-being of humans and animals, and that would require in vivo experimentation. So any US government agencies that were going to be working on animal research needed to abide by these principles. And they're really powerful, and they're very succinct. They're published at the back end of the guide for the care and use of laboratory animals if you ever need to see them. So first and foremost, transportation. This was kind of one of the main reasons that the Animal Welfare Act came into being initially. And the second principle that has so much importance still today with every experiment we think about is that the procedures have to consider the relevance to human and animal health. I love how that's partnered already in 1985, the advancement of knowledge, and the good of society. And this will come into play with some of our most recent proponents thinking about animal welfare. We want to consider the species and alternatives. We want to avoid distress and discomfort. We have to use appropriate drugs, so this gets to refinements, and euthanize animals to limit any pain and suffering. The seventh condition was to make sure that there was a veterinarian involved to help look at the living conditions and ensure they were appropriate. Make sure everyone has been trained to work with the animals. And lastly, decisions of these principles should not rest with the investigators alone, but with an appropriate review group, such as the IACUC and consensus. So again, just building along the 3 R's, we have a lot more detail here to work with, very powerful statements that have continued to leverage how we do our jobs in the scientific enterprise. So again, what was a really important thing that happened in 1985 was that, again, through the Animal Welfare Act, the IACUC came into being. It's 35+ years old now, and each registered research facility was mandated that they had to appoint this committee. Now, at my institution, like many of yours, we have a few dedicated people that have to be on it, and then at Michigan State University, we have up to 20 altogether to fill out the different roles and have a really robust ability to discuss every animal proposal that comes before us. So we have to have scientists. We have to have veterinarians. There are staff that are non-scientists. And very importantly, there is local and community public representation. So the number of things, and there are many that this committee is expected to do, and it has grown in time as the IACUC enterprise itself has become its own business. We have to review the animal activities, ensure compliance, go and look at all the facilities, maintain that training that was mentioned in the US government principles, and look at concerns about the animal care and use overall. Really have complete oversight for the entire animal care program. And it also, in 1985, the investigators were charged to consider alternatives and consult with veterinarians before they would do their work, which was really critical as well. We still do veterinary consults on every single proposal that comes our way. So as I mentioned, the IACUC community really grew and has grown for the last 30-some years. But interestingly, it is one of the areas where I feel like there is some potential misuse or abuse of the 3 R's concept. And there is some finger pointing that happens at the IACUC somehow that they are not evaluating the ethics rigorously enough before approving use of protocols. The critiques that IACUC are not consistent across institutions, that IACUCs aren't consistent in their deliberations, that we are putting members really in an impossible position, I think is what I'd like to state. It is impossible to set standards when the people and the expertise is going to differ across committees. But flexibility was built into the system early on so that consultants and subject matter experts could always be invited to assist in review with discussions. And the last year alone, I have been part of chats external to my institution where IACUCs have been blamed for the reproducibility crisis, that somehow they aren't mandating reduction of animal numbers. But that's a very limited view. And I want to be very clear and share what I thought was such a perfectly stated sentence that captures my feelings. This was stated on a call in advance of this talk today by Sally Thompson-Iritani. She said that IACUC was never meant to be the ethical arbiter of animal science and scientific merit. So why are we and others potentially expecting them to be that final ethical gatekeeper? To me, those who may be placing blame on the IACUC process and the reviews maybe don't really understand all that it entails. And that might be on us to have a more transparent approach to clarify exactly how decisions are reached about approvals for animal studies. I think and believe that this transparency would help not only the scientists themselves, even at our own institutions, understand why their protocol may be being held up for more review. And I think it would really help the public and learn more about the science and especially inform the public that thinks there's no review, that we really do have a robust process in place that considers animal welfare. I just want to touch on this paper at the bottom by Stacy Pritt that just talked a little bit more about this interplay of ethics and how we can think about things differently and how we work for them. There's a lot to learn from this and a lot of discussion still yet to have. So moving on then, there was a very important issue of JAALAS. This is one of the AALAS journals that came out in 2015, and it helped me quite a bit in my preparation for this talk. In particular, this paper at the top by Jerry Tannenbaum and Taylor Bennett, I found it was a very comprehensive and really a must-read for those that are interested in the 3 Rs topic. I spoke with Taylor as well in preparation for this talk. What this paper did was really appeal to promoting a more shared understanding of those original definitions of the 3 Rs in the context of what Russell and Birch had proposed for their new applied science to look at the improved treatment of animal welfare, specifically to improve treatments of lab animals while advancing the quality of science and studies. So the paper dissects the original language, as I said, and parses the intent and impact of interpretations. So they discuss each idea, refinement, replacement, and reduction, as originally intended. And I want to pause here and just comment on the term reduction, because this is the one R of the 3 that I feel is most misunderstood and potentially misused and abused. So to quote the authors of the paper, the 3 Rs principles doesn't define reduction as minimization of the number of animals used, even though that is really largely how we now define it. In fact, Russell and Birch emphasize that it's often impossible to know in advance of an experiment whether the minimal number of animals will be used. They actually emphasized, as we understand now, the importance of statistical methodology, and they were advocating for this new science, as I've mentioned. So I want to demonstrate that the misuse of reduction has kind of become the refrain that I've continued to hear in recent years for those opposed to animal research, when they mention that the 3 Rs, the scientific enterprise, is failing in their goals. If we stop and think about it, aiming at absolute minimization is illogical in the face of experimental design. Animals would be wasted if too few are used, and results then wouldn't be able to be interpreted or repeated. We can also think about reduction if we flip the switch to how expansive it could be, to say that it's important for us to be reducing the pain and the distress and the stress. And maybe that's captured in refinements as well, but it really could also be in reduction. So that's, again, a charge to think about 3 Rs more expansively and progressively in our approach. So this issue, as I mentioned, came out in March of 2015. So the next thing I just wanted to touch on that came out shortly after that, which was really important, again, for how we think about science, was the 4 C's. The NIH coined these, and when studying sex as a variable to strengthen science. We have to consider, collect, characterize, and communicate. So women now account for roughly half of all participants in NIH-supported clinical research, and that is now subject to NIH's policy on the inclusion of women in clinical research. The link here is included for you on this slide. However, more often than not, it was identified that basic and preclinical biomedical research largely focused on male animals and cells from male animals. So this over-reliance on males really was found to obscure an understanding of key sex influences on health processes and outcomes. So it became mandated that females had to be included in certain studies, and considering that, we need to realize that in essence could have doubled the numbers of animals that would be used in different studies specifically, and that the NIH wanted to look at sex as a biological variable as it was factored into designs and the idea of which animals to use in human studies. Of course, all of this still requires strong justification from scientific literature, preliminary data, other relevant considerations, et cetera, if anyone's going to want to use more than one sex. But investigators would be strongly encouraged to discuss their issues with the NIH, program officers, and others prior to submission of applications. But I do think the 4 C's takes a lot of the principles from the 3 R's and expands upon them and makes them relevant as well. So in the concept of the 3 V's, this is another one that came out and was really important to the lab animal field. I just want to mention this was really done as a design by Hanno Verbel to look at the harm-benefit analysis. He believes that in this paper that if studies do not produce results that are scientifically valid and reproducible, then and that is something that we don't want to have happen in the spirit of the 3 R's. So in that case, he was even pointing out no matter how little harm was inflicted, animals still could be wasted with inconclusive research. So his paper proposed that you would think more specifically about design, looking at scientific validity. He breaks it down into construct validity, internal validity, and external validity. And the scope of the animal experiments, his point was, is often quite narrow. We have one laboratory that's working on a very standardized condition and maybe only creates information that can be repeated in that laboratory. So his goal was, by talking about our efforts to minimize as part of the 3 R's reduction, we may inadvertently be exacerbating this limited data output. So his advocacy is really to have repetition of experiments, potentially at multiple laboratories, to try to produce a more generalized outcome. To me, this is fantastic in that we must continue to increase translatability to humans and animals, and this is a great step forward in that. And then moving to the most recent addition to our discussion about the 3 R's, this was a book that came out just last year. Many of you have probably seen it. The authors of it for the Principles of Animal Research Ethics are Tom Beecham and David DeGrazzi, who are both preeminent ethicists. So I've looked through this book. Also had the benefit to talk to Taylor Bennett about an AALAS presentation that Mimi Rogers and Margaret Landy put on, specifically looking at this new ethical framework and applying current case studies into the 6 principles. So in short, what they discuss here are 6 principles, 3 of which evaluate social benefit to humans compared to principles of animal welfare. And there's not much as far as the explanations behind these. I think it's really interesting. What I really like about the text is that there are a number of chapters that they've invited authors and well-known professionals and colleagues to come and share their stakeholder perspectives. So there are veterinary medicine perspectives, biomedical researchers, psychologists, lawyers, and bioethicists, that, including lab animal vets that are involved in this as well. And it offers a framework that bridges a gap, in their opinion, between the concerns of both the biomedical and the animal protection communities. So there's a lot of press that came out around this, but importantly, I want to share some words from Mimi and Margaret's presentation. These 6 principles were not designed to compete with or necessarily replace the 3 R's, although there was quotation taken from the authors that they felt like the 3 R's were limiting, and this was the next step forward in their mind toward thinking more holistically about assessments. So they reflect maybe a little too much depending on that classic or limited interpretation of the 3 R's. But I would in fact say that in the contemporary 3 R's world, we would want to include these in our ongoing evaluation. They're just kind of another step down the path as we continue to evolve our thinking. So for some artistic break then again, we've got what I would propose our progressive 3 R's. So in my umbrella analogy with hands held underneath in community really, the umbrella will take on the 5 freedoms. It will include our 9 US government principles. It encompasses the 4 C's, the 3 S's, the 3 R's, the 3 V's, and the 6 principles. So in a progressive state, as we brand this, it's an acronym that covers and includes all the successive and related concepts. And I really, again, want to point out that the stakeholders, everyone, all of the groups that are involved in animal research should discuss aspects of the 3 R's holistically to work through the ethics and discuss the benefits. So one final thing I'd like to address in our last few minutes together is just about the animals themselves, because this can often be a critique. Somehow in our very intense focus, the NIH focus on scientific efforts and reproducibility, are we being attentive to the animals themselves? So there have been and continue to be advancements in the care and quality for animals. These are mainly conducted by caring, devoted professionals who want the best quality of life for the animals. And that makes good sense because we want to have enriched and balanced animal lives that then provide really robust scientific data. Critics of the 3 R's often cite that there's an inherent harm to animals, and that's a fallout of limited ethical review. However, to that critique, I feel like we have to respond with data and facts and show the information that's been gained in veterinary science and the involvement of veterinarians in virtually every aspect of contemporary animal use for research purposes. So even just starting with something as benign as the oath that veterinarians take upon graduation highlights the fact that we are charged with advancing medical knowledge, along with making sure that we are keeping the principles of veterinary medical ethics at heart. In addition, as mandated and as I mentioned earlier, there are the requirements of veterinarians on committees, being involved in the review. One or more qualified vets must be involved in the review and must be involved in the IACUC. So we have the authority to oversee the vet care program and provide oversight. We help with things like disease prevention, diagnosis, and control. We have to be available and look at animals 24 hours a day. We promote animal well-being, and we're also adhering to any of the methods that might come out from the AVMA, the American Veterinary Medical Association. So we have a changing landscape in the idea of animal research. Landscapes in theory, this is Van Gogh's wheat field, is based on color theory. So there's different colors next to each other, and how you look at them alters the eye's perception. The strokes themselves are expressive and sort of take on a physical presence of their own. That was a style that Van Gogh had. So yes, if you stand back and look at it, it's a wheat field. But it's so much more when you assess the colors throughout. And the individual strokes make the difference in the beauty of this. So I wanted to quote an analogy from my colleague, Dr. Melanie Graham, who I've been working with on a project about the 3 R's, who's also going to be presenting at this conference. She is a very well-known scientist who works with non-human primates. And what she shared is that the animal welfare that we're talking about isn't necessarily a broad concept. It really happens at the individual animal level. And that's what we're focused on. She notes intrinsic links between welfare and scientific outcomes, that well cared for animals lead to increased benefits of data outcomes and, again, more translatable, relevant applications. So just to speak to this a bit, there's so much we could talk about related to animal care and so many species here. Of all of the pictures that I've pulled from the internet here to show you, there is an emotional response, sometimes a physical response when we view these. We think about how these animals are like us. We think about how they empathize with us. Interestingly, work from Jeff Mogul's lab has shown that rodents are empathetic to one another. They have emotional contagion. We need to learn more about their intelligence, Frans de Waal has done amazing work talking about how smart animals actually are. Elephants work together to find food. An octopus has been shown to be as intelligent as a kindergartner. Birds can make tools from objects to build nests and things like that too. I mean, it's really, really fascinating. And we're learning all the ways in which opportunities can be provided to assist with expression of species-specific behaviors. This presupposes that we have insight into what those behaviors are, which may not always be the case. But some recent lab animal science information has shown that even just giving enrichment to different species will allow them to modify pain behaviors and diminish pain more rapidly, particularly in rodent models. We've started the mandate for social housing for social species, as this is a key welfare technique, but one that requires veterinary oversight because some social species, as you are aware, will fight one another to establish dominance. So the behavioral and the environmental management techniques that ultimately allow animals to adapt, we have to learn about those physical and social conditions. And yet we're so pressured by the current societal culture to just continue to reduce numbers and replace animals. We have to still study animals for the sake of the animals themselves. It's a key consideration. So moving on from this then, I'd just like to share again back to our progressive 3 Rs. We will have instances where the animals then also fall under the umbrella. They will always be under consideration. And I think, again, in the progressive model here, we have plenty of room to consider the animals. Now, I mentioned the progressive 3 Rs to you. It's a terminology that's been coined that also could be synonymous with contemporary. And I don't want to leave out crediting the NC3Rs site and the committed individuals that work on thinking about this same concept. As I mentioned in the beginning of the talk, I am just building on what others have done before me, standing on the shoulders of giants in this regard. So the contemporary thought would be, again, we don't limit what we think of these 3 words to be. We're looking at them in much more expansive interpretations, and I would encourage you to look at the NC3R site to see this. So for veterinary medicine, we are doing research, and it is scientific research, and it's looking at all kinds of items related to disease, therapies, how to abide by refinements, looking into animal health, knowing about their behavior. This is all a very rapidly expanding science, and we translate this into practical information that's critical to continue to minimize pain and suffering and also ensure robustness and reproducibility for experiments. So I just want to point out that this in and of itself, the idea of veterinary management of animals, will continue to be a massive area of discovery. There are a myriad of veterinary journals, animal science journals, professional seminars, et cetera, that are looking at this specifically. And these are evidence-based improvements. They are based in fact. We are in a very unique time, as we all know, and the pandemic itself offers a great example for how to think about animals. The idea that we need the right model for the right question. It's the scientific process itself that's going to drive our ability to ultimately reduce animal numbers in certain disciplines. For example, the COVID vaccine, that a lot of people have said, oh, well, we didn't need animals for that. We just got it out into humans as quickly as possible. They haven't got all of the data. They haven't got the information that a lot of the work was done in animals for SARS several years ago and now has been able to be reused and adapted more quickly from animal work that was done in the past. So we do have an ethical obligation more than ever to keep driving science forward and create new knowledge. I think this still goes back to principle number 2 from the 80s. Procedures should consider relevance to human and animal health, advancement of knowledge, and the good of society. Just mentioning here, the harm-benefit assessment as it underlies everything that we do. So a couple of quick things as we finish up. There has been a pandemic pet proliferation. There's a lot of alliteration there, but I thought it was really important. This article just was shown out a week ago in the Michigan Veterinary Medical Association, and I know this personally. I have not acquired new pets, but I have so many friends and colleagues that have. And so when we think about this, we are not reducing animal numbers, we are creating animal numbers. Not only adoptions from shelters, adoptions from research institutions, but also from breeders. This is what's happening because science shows that pets and humans have incredible health benefits when there are times of stress. We've known this, but again, this is a field of study that involves animals, social sciences, and human work. So when we think about proliferation, it was timely then that just last week, in the last couple of weeks, there's been a big splash about how many animals are too many. Where does it depend on? Is there societal benefit to one but not another, and who gets to decide these things? So I wanted to just touch on the fact that this article came out basically stating that there's 100 million animals. It was based on a paper that was put together by Dr. Larry Carboni, who is a lab animal veterinarian. He made some estimates based on data that he was able to get through conversations with colleagues and through some FOIA requests, where he estimated that there were more than 100 million mice and rats being used in labs today. And he acknowledged it was just an estimate, but from that, there were, in David Grimm, who's a science writer, he sort of pushed and I think maybe glossed over some of the important statements that Dr. Carboni placed into his publication. So he acknowledged that it was just an estimate, and he also stated that that was about 99% of all the lab animals were rodent species. He also pointed out that there is a system of inspections in place, that we do whistleblower investigations, that there is coverage and oversight for rodents in public and private institutions. And those of us within the animal biomedical community and who participate in all of these activities are well aware of these oversight activities that I've discussed. The IACUC review, the vet consult, justification of animal numbers, and alternative considerations. But unfortunately, in this summary, I felt like Dr. Carboni was glossed over by Mr. Grimm on these important statements and kind of tilted the angle to animal protectionists. There wasn't the facts, there wasn't accurate data, everything was based on estimations. And he did acquire a quote from the president of Alternatives to Research and Development Foundation, which their goal is to reduce the number of animals in labs and find replacements. And her statement was, the figures are troubling because mice and rats aren't covered by the Animal Welfare Act when we know they're covered by other regulations. And her quote was, if the numbers are anywhere near correct, the amount of pain and suffering that's occurring in these animals is acceptable. Myself and others find this to be like really sort of reckless in a statement. There was no information provided as to the care and well-being, the models that these animals were used for, the oversight, the interventions. So I just want to echo my colleague, Dr. Allison Bennett, who was also quoted in the Science Mag article, that she also agreed it was a pretty disappointing analysis. We have to think about things more broadly, and we have to have more discussion about them. So thinking about that, if we were to reduce animals, another area in our case studies for now includes what type of rodent we're using. So the microbiome has been a very hot topic. The microbiome is basically the full set of flora that we have in our gastrointestinal tracts and the interaction between them and our brain and other systems. And so wild mice, which normally many would consider to be pests, there are whole businesses built around getting rid of invasions of wild mice and wild rats in our communities and our homes and our farms. Now there's going to be more of them used in research analyses. So does that type of rodent, what we use, does that matter to society? This will be an interesting and ethical discussion ahead as well. And the last thing that I wanted to point out, which I just thought was really just a super cool article, was from the New Yorker last month. It talks about, and I think is a good example of the progressive use of the three R's. So CRISPR is commented upon here. I mentioned that that this is a 2020 Nobel Prize winner as a technology. Jennifer Doudna was one of the recipients from UC Berkeley of the prize, as she's one of the developers. And she calls it a way to rewrite the very molecules of life in which we live. So interestingly and briefly, this article talks about how CRISPR has been used to manipulate the cane toad, which is overtaking the continent of Australia. It was introduced to basically try to control pests like bugs and things like that, but evolution pushed quickly and these cane toads grew very large. They're 6+ pounds each, and they have just taken over. So gene editing using CRISPR is now available to change the skin tone of the toads so that you can identify which ones have been manipulated. You can also impact the fact that each toad can give birth, can lay like 1.5 million eggs. Explains the proliferation. The problem is that if we manipulate the gel layer on the outside of the eggs, they may not be able to be fertilized. When these toads get big enough, they will eat anything and everything that's smaller than them, including other smaller cane toads, snakes, alligators, etc. So in this idea of the progressive 3Rs, we need to think about reduction, using science to reduce the number of cane toads to allow the proliferation of species that otherwise are bordering on the brink of extinction. So the goals here are to enhance biodiversity through genetic rescue, and just kind of a fun story. It's social benefit versus animal welfare. So these are the discussions we continue to have. So for the future 3Rs, I would challenge us to think we're going to continue to have more principles that come into play as people talk about it further, and we're going to have more species like the cane toad that we have to discuss. We are all advocates in this process, every single one of us. If we're talking about it, we have an advocacy investment. And so what I just wanted to point out about the importance of that is that we want to take what we've discussed here and make it better if we can. There are so many advocacy organizations. I'm going to list a lot of them at the end of the talk. And we need to have this advocacy to safeguard the future of biomedical and veterinary research involving animal models. It would be naive to assert that the only advocates for animals are those in the animal rights camp or in the animal protection groups. Our whole community drives momentum toward animal advocacy, and we have to continue to do that for beneficial outcomes. One comment, I think many of you may be aware that there is a working group that's ongoing right now. I've had the privilege to be involved with that through the NIH. This was charged as a group in the late 2019 to come together and talk about assessing and making recommendations for reproducibility and the rigor of animal research, specifically to improve experimental design, optimize translatability, enhance training, and increase transparency. These same themes that I have been talking about and showing you have been in discussion since the 1970s. So we're continuing to look at gaps. The report should be coming out in June of 2021 here. We're still having meetings about it, and it will be something to look at on the horizon. So the 3Rs are a masterpiece. Just as I'm showing you here, these are self-portraits that Van Gogh did. Initially, they were not regarded as anything special, and over time, they have become viewed as masterpieces. And what I wanted to show you here was the progression in his work. So from left to right, Van Gogh started off as kind of a country, living in the country with his family, and then he moved to Paris, and he finally paints himself as an artist. You can appreciate that there's a change over time, and it is a triptych, again, like our 3Rs. The 3Rs have remained ubiquitous in large part because of their benefit to the animals, but they also have importance for us as well, the stakeholders. So here is the icon that I've put together just to express how we could reconsider the foundational or classic 3Rs with different interpretations, very much combining them with the synergy of other ethical tools of analysis and validity assessments that we've talked about. All of these are going to be essential in the ongoing intricate considerations of the appropriate and future animal use. So in artistic terms, the 3Rs themselves are also a masterpiece, and they're a work in progress. And I hope we can all appreciate that and continue the discussion. So with that, I'd like to thank the organizers for the LabRoots conference. Had a lot of really fascinating discussions, and we'll continue to do so with the colleagues here from Lab Animal Medicine and Priminar. If you want to take any arts classes, I thought I should just do a little plug for the Barnes Museum, and all of the advocacy groups are listed on the right, many of which I'm a part of and many of which I've learned from. So thank you very much, and I really hope you enjoy the rest of the sessions.